have 5 minutes. If you can finish at time 15, you have 5 minutes for questions. If you can't, you carry on until 20, and then there will be no time for questions. Okay? So, yeah, that's basically the plan. And one after the other, so there's no time, yeah, only during the intervals. Okay? Let's wait until everybody. Where's the third person? Yeah. Unfortunately, Florencia had a problem with a family there in Buenos Aires, so she had to return on Sunday, past Sunday. It's unfortunate. But she's participating, no? She's here. Mm-hmm. Hello, everybody. For the ones who are not here, who are watching via YouTube, welcome to the last day of LASCON 2026. Today is the student projects presentation day. We have 22 projects presented by the students, starting at 9 a.m. São Paulo time, so in 2 minutes time. I'll be enforcing that time is really kept as it was announced, so 20 minutes for each presentation, 15 minutes for the proper presentation, plus 5 minutes for questions. People who are watching and want to ask questions, unfortunately, I think it would be difficult for us to manage that, so only people who are here would be able to ask questions. But the ones who are interested could send emails to me, and then I forward them to the students, because it would be difficult in 20 minutes to manage this. Okay, so I'm sorry for that. So that's about it. So we'll start in about 1 minute, just to keep time. As you know, we in Brazil we in Latin America like to be really punctual. So yeah, of course. Yeah. So yeah. I'll let you know. No, I'll let you know when we start. Let's start on time. Okay.
Yeah, okay. So good morning, people. There's a lot of people here we weren't really expecting, but thank you for coming. Today we're going to present our LASCON project that our group is composed by me, Cunha, Pedro Muradas, and Florencia. And yeah, our title is The Impact of Structural Topology on Informational Theoretic Characteristics of Spiking Neural Networks. Big words, right? But basically, we investigate how different topologies of a network can maximize a informational processing, right? So, yeah, and then let's begin. Basically, the idea of our project comes from this evolutionary trade-off that biological neural networks face, which is that basically there has to be a balance between functional specialization that favors local connectivity and computational efficiency that favors long-range communication. So if this balance doesn't happen in the brain, for example, you could have impairment in the integration of the brain regions, for example, or even pathological synchronization, such as in epilepsy. So it's important to keep that balance. But this is not just a biological issue. This is also a computational one. So modern AI systems rely on this dense all-to-all connectivity, which is very it has a high energy consumption and could lead to these representational collapse where these local redundancies of informational in this local connectivity loses its informational to this global randomness that they could have. So yeah, this is a trade-off that these systems have, and what we try to do is to solve it, basically. So our hypothesis is that in a small world topology of a network, we will have the maximization of informational processing. So the small world topology is this middle one. We increase this p variable, which is called rewriting probability. A low p value is a more regular network, more regular connectivity, and a high p value equals 1 is a total random network. So what we suggest is that it has to be an intermediate state between those two, a small world topology that maximizes the informational processing. So what we did to investigate this, we simulated this network with Izekevich model in Brian 2, which had 1,000 neurons in 8 modules of 100 excitatory neurons in these 8 modules, and one pool of inhibitory neurons of 200. So basically what we did with this network, we tested various topologies that were using this Botz-Strogates rewriting process, which basically is very simple. You basically varies this p that you can see in this image, which is the rewriting probability, the probability of one neuron being connected to another neuron in another module. So yeah, that's what we did. And here in this right side, you can see different topologies that could be like module ones, module configuration to total random configuration. And yeah, that's what we did. And here you can see some sample runs of this network that basically follows what this rewriting process is. You vary the p, so you get these spike trains that has this winner takes all effect, where you can see like one module more active than the others in some intervals, right? So it's a winner take all of thing. And then as you increase p, you increase the randomness. So the redundancy gets higher, and you see this synchronization between the modules. But yeah, the winner takes all situation, and here the total random synchronized situation. And yeah, and what we did also about the methods is that to analyze how the informational processing was being, how it was in each of these topologies, we calculate some informational theory metrics a specific metric, which is called transfer entropy, that Pedro is going to talk more about. And yeah, so.
Okay, so now that we have established that we have a motivation that both biological and artificial systems rely on this integration and segregation trade-off, we are now going to investigate how the topology and how we measure that in different topologies. So now I'm going to describe how we formalize this computationally. So as Carter mentioned, we have this modular independent activity with when p is equal to 0, and as we increase it, we have a more oscillatory and synchronized regimen. So apart from what you can see visually, we can also sort out some aspects about the information as well. So for example, here we have specialized information that could be, the information can be processed locally, which can increase the amount of specialization. But here we also have some redundancy. So we have a lot of trade-offs with information theory. And how we decided to approach this was using transfer entropy. So transfer entropy is a metric that calculates the amount of information that is being transferred from one module to another one. And how we calculated that in our project was by using the spike counts for each module. So pretty much what we did was that we calculated the spike trains, simulated the spike trains for each neuron in each module, and then we summed the spike trains of each millisecond in each module. So for example, if module A has 3 spikes in 1 millisecond, then the time series at the end will have a number 3. So it's a non-binary discrete time series for each module, right? I think that's, that's understandable, right? So then we generated a time series, that one for each module, and then we used those time series to calculate the transfer entropy. And there's a lot of ways to interpreting it and analyzing it, but one that I really like is this one where we have the diagram. So here we can see that the transfer entropy is pretty much the amount of the amount of information that is being transferred from module X to module Y. And how we do that is that we calculate how much of the uncertainty of module Y in the present state is reduced when we know the past state of module X, right? That is already explained by the module Y. That's pretty much it, and that's why we use entropy. Right, so obviously we do have to assess some statistical significance and control for spurious information transferred. So we constructed surrogates data sets using circular time shifts. This type of surrogates preserve the whole temporal structure of each spike train, including the firing fluctuations, the statistics as well. So for example, interval spikes, CV as well, and also autocorrelation. So, but it selectively destroys the interneuron causal dependency. So this is very great because we keep all the statistics in the simulation the same. The only thing that we change is the temporal dependency of one series to another series, right? And this is kind of like a big deal because transfer entropy is highly reliable on the temporal structure of the data. So if you use a naive shuffling, for example, you would artificially reduce intrinsic dynamics anyways, and therefore you would generate a biased null distribution. So anything would pass the comparison and the p-value wouldn't mean anything exactly. So the circular shifts therefore provide a more conservative and biological meaningful node model, isolating directed interactions while keeping each neuron's internal dynamics intact. So the Python library we used was NumPy, and we literally just used like the roll, because that's exactly what we do. We just roll the temporal series, and then we calculate the null distributions. And the way we did that for each pairwise comparison, we generated 350 surrogates of circular time shifts, which yields for us a 0.002 of threshold of significance, which gives us more than enough confidence to reject or not the null hypothesis of p-value for 1.5, right? So right here. And well, that's pretty much it. So now let's go to the actual results. Can I use this? Yeah. So this is like one of the main results, the results that we have. And what we found out was that we found a strongly non-monotonic correlation between the transfer entropy and the rewriting probability. So here we have 100 trials of the 100 neuron networks that we found. And here we can see that we have an increase of transfer entropy as we transition away from this regular independent module network. And then we arrive at its peak at around 0.6. And this is very interesting. And then it decreases as we approach a more random graph, a more random topology. So before I talk about the p-value itself, it's very interesting because we found a more than 50% increase in the effectiveness of information flow. This is very interesting because it's not just a subtle amount of information that is being enhanced. It's actually more than 50%. So it was quite impressive that. And also, I do want to mention that it's interesting that the point of maximal information theory and that the transfer entropy was actually in the middle, neither in the completely random modular independent type of topology or the completely random all-to-all connected, because we have high redundancy here and we have low communication here. So here we see that the middle ground is actually perfect because it enables a modular structure, also some shortcuts that enable global integration of the data itself. I'd also do want to assess that we have some low kind of increase in bits. If you think about it, it's 0.01, but if you actually, it's actually 0.1. And if you actually dive into the data and the literature, we actually see that this is extremely high for transfer entropy calculus, because usually the values lie between 10 to the minus 3 and 10 to the minus 1 in increasement. So what we found was actually a huge increase in transfer entropy for these kind of metrics.
So yeah, in summary, we addressed the fundamental biological and computational trade-offs that I was talking about between wiring costs, modular specialization, and global integration. We did that by systematically varying the network topology, the P rewriting probability that we talked about. We showed that direct information transfer is maximized in a small world regime, just as Pedro was saying, at the transition between segregated modular dynamics and globally cooperative dynamics. And this provides a mechanistic explanation for why biological brains exhibits these modular small world architectures under metabolic and wiring constraints, the trade-off I was talking about, and suggests that dense, unstructured connectivity in artificial systems is not computational optimal. So also in computational systems, this could be extrapolated, right? And yeah, instead, in structured and space connectivity with this strategically placed long-range shortcuts emerges as a principled design strategy for scalable and energy-efficient neuromorphic and artificial neural architectures that are really like famous right now, right? And yeah, and also I think that we did was we formalized this project in a paper that we could share after it's been written yet. So yeah, and that's it. That's it. Thank you so much. Yeah, here we have the emails if you guys want to ask any questions, or you can ask them right now. And yeah. Yeah, a small world topology.
Yeah, we kind of have that as well, because in each module, the connections, the local connection inside each module are randomized. So locally, very locally inside each module, the connections are random, but at large scale, they're not. So within the modules, yeah. So that's pretty much similar.
Oh yeah. exactly the connections between one module to the other. So this P here, we didn't vary any other. Um, yeah, we did in the network itself, right, to test if it would behave just like we showed here. So to get here, we had to change some parameters that are not these ones. We have to change what we did. Yeah, like the weights of the network, this weights here, we have to change to get that winner takes all, for example, in the P equals 0. So, but the mainly thing that we varies was this P, because if you change everything and then you can compare, right? So, yeah. Yeah.
That's true. That's
Yeah. Sure, sure. Yeah.
And the ratio of inhibitory and inhibitory neurons is 80-20, but we've tried for 70-30, and pretty much anything other than that fall into either a coma without activity or epileptic state. So these are pretty much the final results.
Second microphone.
No, I think Antonio it's meant. I need to keep the neurons should decrease the weight of excitatory synapses, otherwise you get epilepsy or So it would be important to investigate whether it is an effect of the topology, topology itself, or an effect of a balanced net or something.
Yeah, there is. I mean, the first 2 weeks we're just trying different parameters, and we found probably 1,000 curves like that, and some that didn't look like a Gaussian distribution. So it's
Good morning to everybody. Good morning to everybody. Today our group will present our project entitled Domain Modulation of Astrocyte Calcium Dynamics in the Tripartite Synapse. So this presentation will be divided in these parts: the theory, objectives, equations, methods, results, and discussions. And to start, I will make a brief introduction, especially focusing on the biological fundamentals before entering the biophysical and the equations. So to start, we must know that traditionally we know that the synapsis is known as a neuron-to-neuron process, but we must know that there is an important part which always participates, and is the astrocyte. So we will try to separate this. We know that primarily the action potential reaches the presynaptic neuron, and then this releases the glutamate in the synaptic cleft, and the AMPAR and NDR receptors of the postsynaptic neuron are activated, and they then translate it in the synaptic transmission. But the astrocyte has an important role in this type of synapses. Why? Because the astrocyte has receptors which receives the glutamate, and then it activates the calcium intracellular production, and then this produces a glutamatergic output, which will be translated in the slow inward current in the postsynaptic neuron. So why this is phenomenologically and biologically valid? It's very important to know that, for example, in this picture, we can be the IP3, and the IP3 has an important role. Why? Because the dopamine and the glutamate doesn't have a direct role in the calcium output in the astrocyte, because they first must try to modulate the IP3, and the IP3 will also try to modulate how many calcium volume will be produced by the astrocyte, and then this will be translated in a slow inward current in the postsynaptic neuron. So we have that glutamate and dopamine have different roles. They don't assume, while the glutamate as primary driver of astrocytic calcium signaling, the dopamine has the role to modulate all this process. The glutamate is something primary, but the dopamine has the principal role to modulate this. So, for example, if we just use glutamate or only dopamine, the synaptic signaling will be different. So the objective of our project is to investigate how dopaminergic modulation of astrocytic calcium IP3 affects the synaptic transmission, and also how this IP3 production will also affect the glutamatergic input and the astrocytic response.
So for the mathematical equations, we decided to work with this classic model that was the one that Marjalina exposed to us. So basically, it is the model of an astrocyte. We have 3 items in total. The first one is about cytosolic calcium concentration. So here we have a term that refers to the release of calcium from the eye, and also we have another terms just like noise, leak, and pump that is referring to a dumped factor. And also for the other item we have IP3R inactivations, in the sense that this is a feedback system that when has an excess of calcium will turn off the release of calcium. And also we have an IP3 concentration that is the base of the project, of the behavior of the astrocyte. And basically the most important aspect here is that IP3 has an initial state, and it will has a gain over this state from a Poisson distribution that in this case it is implicit, like it is implicit like glutamate here. So for the tripartite synapse, we have 3 aspects. That is the reason of the name. First, we have a presynaptic neuron. So this one will be realized all the excitatory pulses that as interpreted as glutamate here. And also we have the astrocyte that will respond to that as an IP3 and as a calcium oscillation. And for the postsynaptic neuron, we have a slow inward current that basically it functions by the threshold of calcium, and that means that all will be produced if the threshold is overpassed. And that was the problem here, that was to include dopamine. So we have a reference that was a compartmental model, And here the dopamine, it's modeled as a sustained signal that will also depend of the presynaptic spike. But if we include the dopamine in this way, we will have two drives and we will break the logic that the basic model that we exposed before is talking. So for that reason, we decided to modulate dopamine as an event in a continual signal of a pulse. Here we have the methodology. So basically we use MATLAB and NEST for the simulation. And the final equation, we have an IP3 current that will be current with the biological behavior that we are seeing in the Rancel models.
Okay, so now we are going to talk about the results. And first I will present, this, the model, so the system. And in our tripartite synapse, you have postsynaptic and presynaptic neuron. And but the model we choose here as a neuron is the ADEX with consistency-based excitatory synapse, where we choose in the spiking, the parameter that we choose are for the spiking activity regimes. And we fix the threshold for spike 0 millivolts for the membrane potential. So, and here we decide that this presynaptic neuron will be stimulated by external current, and we can choose when we can apply the stimulus. And as the astrocyte, we use the Lin-Riesel model, and in that model we add, as my colleague said before, we add the glutamate. We will take into account the glutamate and the dopamine. The glutamate will be released by the presynaptic neuron upon spiking, and the dopamine will be modeled as a discrete event from external sources. That means we can add the dopamine only if the presynaptic neuron is spiking. If it's not spiking, we can add this dopamine. So, and the equation is that's one. Now we use first the MATLAB software to implement our model, and using the Hunch-Coulter, 4 other Hunch-Coulter method for integration. So first we are going to present the baseline versus dopamine in tripartite dynamics. I mean, first we simulate our model without adding the dopamine. So, and in this figure 1, we observe that with a baseline, a considerable baseline of delta IP3 glutamate, yes, we observe that the astrocytic calcium enter in transient regime. So we observe here many oscillation of the calcium dynamics, and in the lower spaces we observe that the presynaptic neuron, when it's spiking, the postsynaptic neuron also spike at the same time. So in that case, they are synchronized. So now when we add the dopamine, he increase or modulate the calcium astrocyte. And here we observe that the seed current is more strengthened. I mean, the amplitude is more than when we didn't add the dopamine. Supporting that the dopamine here is play as a gain modulator in the tripartite synapse. So to generalize this, we try to variate both the delta IP3 glutamate and delta IP3 dopamine, and we observe this 2D diagram. And we observe that when both the fraction of glutamate and dopamine are less, we have subthreshold oscillation. The calcium astrocyte didn't oscillate. But when both are considerable high, so then we observe a high oscillation or saturation. So here we count the number of times the calcium crossed the threshold. And here we present some illustration with some point we took on this two diagram. Now, given that during the LASCON lecture we have learned the NEST simulator, then we try to implement this model into NEST simulators and try to see if you get the same result. And our hypothesis is confirmed. Here we maintain so the presynaptic input and the glutamate, but just vary the delta arbitrary dopamine. And here, when the rate is low, we also observe that this dopamine modulates the calcium oscillation. So, and here we increase the rate of the dopamine release, and we observe that the same phenomenon, and so it modulates the calcium oscillation. So in conclusion, we can say that the dopamine alone doesn't trigger the calcium activity or oscillation. But when it is added to the glutamate, even if it is low or small, yes, this they can combine and enhance the or make the calcium enter to the oscillatory regimes, and then increase the impact of the C current to the postsynaptic neurons. So as a limitation of our work is that we use only unidirectional connectivity. I mean, we didn't include the feedback from the postsynaptic neuron to astrocyte, nor the astrocyte to the presynaptic modulation. So as we know, they exist in biological tripartite synapse. But nevertheless, our model simulation support our hypothesis that the dopamine acts as a gain in the calcium or as a gain modulator in tripartite synapse. So this is the reference we have used. We thank all the LASCON lecturers, and thank you for your kind attention.
5 minutes, questions? In your model, you have this, you say that dopamine. What specific effect you have that's related to dopamine instead of any other neuromodulator, like norepinephrine or any other type of modulator that acts in the nervous system? Why, I mean, do you choose dopamine instead of other? I mean, is there any specific reason why you chose dopamine instead of any other? To make it more specific for dopamine, say like having receptor specific for dopamine instead of any other.
we just decide to study the effect of dopamine. We did take into account another modulators in the, the in the brain. So and and here we decided that the dopamine can be released by another cells instead of the presynaptic neurons.
No, I'm just suggesting that you could make it a more general model. So you just say that any neuromodulator without singling out dopamine. You could just say that you have the glutamatergic effect and you have a neuromodulatory effect without mentioning specific dopamine. Could be any neuromodulator, according to what I understand from the model.
In this case, because we use some specific reference, we decided to kind of replicate the behavior that we were seeing, for example, in a compartmental model, taking into account that this is a really not complex model. So for that reason, we decided to use dopamine and not another modulator. But I mean, in the logic that you're proposing, it could be any neuromodulator. So yeah.
And also because from the application of this project can be that a loss in dopaminergic activity can cause different disease like Parkinson's. So maybe this is a focus that we can try to translate.
It's just a question for my general culture. I know that and you mentioned Parkinson and indeed dopamine acts in the striatum, and we know that some of the action is presynaptic. Is it what we are talking about here? Is it the presynaptic effect of dopamine is acting at this tripartite synapse, or is there another presynaptic regulation of the dopamine? I just don't know the answer. I'm just asking. Do you know?
Here, as we said, from the literature, there are many. We have read one work, which assuming that the dopamine can be released by the presynaptic neuron, but here we decide that it can be released by another cells, not the presynaptic neuron. So that's why we add that as an external another input to the IP3 equation. Site. And as I mentioned, if the presynaptic neuron here didn't spike, so we can say that the dopamine can be released. It's in that interval that we release the dopamine.
So, good morning. I am Gabrielli, this is Solange and Anna. Our work today is about informational thresholds and pathological forgetting in associative memory networks with synaptic decay and unlearning.
Okay. Basically, we use the hot field model for all of our simulations. And well, in this model, we have stored patterns that with the mu that goes from 1 to p, the number of patterns, and we have a that is a neuron that go from 1 to n, the total number of neurons, and basically we have to activate values that are +1 when the neuron is firing and -1 when it's not firing. And well, the dynamic that we have is this one, where we multiply a matrix of weights with the pattern and add a noise, a Gaussian noise in our case, and all this goes inside a sine function. So well, basically, this model has this space of memories where the storage memories in the matrix that is constructed with the Hipps rule, where you multiply the patterns, and well, each of these patterns that are stored is an attractor. So if you start in a place like here, you are going to go to the storage memory, or well, that's what should happen. And so well, what we want to study is memory loss. So we add different mechanisms to make the network forget. And well, one of the models that we propose, based on different literature that we read, is using a weight decay with an exponential decay, basically, where all the weights are degraded in the same way. The other model is the synaptic loss, where we use a probability of gamma to set a weight to 0. And we did this on a symmetric way. And well, the third way was using an anti-Hebbian term, that is this here. And on this way, we wake the stronger patterns that are on the matrix.
So the objectives of our work is to use Hopfield model as an associative memory network, implementation of the model using terms that can represent the phenomenological forgetting, and looking for an information threshold based on phase diagram that can separate the different dynamics. So the methodologies and results are, to start our work, we did some network using the Hopfield model and habit theory, and we were inspired in this work of Kinochi. We implement some patterns with different strengths. The strong, some patterns were defined as strong, that has deeper bases of attraction, and another patterns were defined as normal, which have moderate stability. And this was the key of our project, because without this we cannot see the pathological retrieval. So we implemented two anti-Hebbian mechanisms. The first one was eta F, that was fast and reversible, and the second one was eta S, that's slow and irreversible. And this term was the one who weakens patterns that are too strong in the network. And in our simulation, we onset the slow learning at the time equals 1,000 seconds. So the first results we obtained were this graph, where eta S is equal 0, and we can see the strong patterns dominates all the dynamics all the time. And when we implement the eta S greater than 0, we can see that the overlap of these strong patterns decreases gradually, and we can assess the normal patterns along the time.
So well, and well, continuing with the other two models. Well, basically we have the difference within them are that in one we have an homogeneous mesh distribution, while in the other the distribution is random, because we are killing a random weight on the matrix. Also here, well, the network topology is going to be altered, while here is preserved, and well, we have two parameters, the time and lambda, to modify, while here we just have the gamma, that is the fraction of synapses that are removed. So well, what we did is analyze how the network works when we stretch the pathology. And well, in the case of the exponential decay, obviously as we make lambda bigger, we see that the mutual information tends to 0. So well, the network starts to forget the patterns that learned. And also, as we increase time, the decays start to be bigger, faster. Well, and in the synaptic loss, and well, also as we make gamma bigger, we start to lose more weights on the matrix, and well, we have a decay also in the mutual information. So well, then what we did is well, use the hot field model, but well, we need the learning gain. So basically, in the hot field model, we have the neuronal states as well, then we have the store patterns where P is equal to alpha n, and alpha is the capacity of the network. And well, we have the synaptic ways where we add this term here, the A, that is the learning gain. And well, the neuronal dynamics that I already explained, and we measure two things. One is the overlap, that is the multiplication between the pattern that you are seeing and the other patterns that are stored in the network. And well, we also calculate the mutual information, that is 1 minus the entropy, and the entropy has this formula. So well, we basically use this model, this initial connection that I explained, and add a weight decay, the exponential decay of all the weights, and also add an anti-Hebbian term after this to the matrix.
So well, for us to start and actually analyze a phase diagram in which we can see the informational thresholds, we have to like analyze the Hopfield model itself, and we can see there that we have two states, which is one is a retrieval and the other is a de novo retrieval. So basically, if we are in a state that it's near a pattern, we basically go to the pattern if we are in a retrieval state, and we don't go to the pattern if we are in a no retrieval state. And adding all of these things that the girls had already told us, we can create a confusion state in which we are going to see what happens. But basically, for us to do this, eventually we have to basically determine two parameters that are going to be the independent parameters. One is the overlap and the other the mutual information. And with this, we're going to set ranges of eta, which was the constant related to the underling method, the lambda, that it's the decay, the exponential decay, that is going to rule the exponential decay, and the sigma square, that it's the related to the variance of the noise of the dynamic itself. Okay, so basically, for us to start this analysis, we set a critical overlap, which is going to be 0.1. This is based on the overlap with all the parameters that we have in our network, and the critical mutual information that is going to be related to only the pattern that we want to go, okay? And that is 0.05. So based on those numbers, we got to these ranges, and we are going to see these three stages that we have mentioned before, for the from the noise and the eta and the lambda that I have mentioned. So with this, well, we take the weight decay and the unlearning rule, both apply to the model of the Hopfield model, and we see these three stages. And here, we during this simulation, we respect the ranges. That it's for eta, it's 0.0, and for lambda, it's 0.0 also. So if we don't go through these ranges, if we go like a little more, we don't see the 3 stages. So that's why we choose for those one. So if we only see the healthy retrieval, which is in yellow, and we have there an overlap that is greater than the critical overlap, and a mutual information that is greater than the critical mutual information. So what we see here, and Annie has already told you, if we are in a state there, we go to the pattern that is near this state. Okay, so what we see in the dynamics of each of the neurons in the network, we see that over time, the pattern doesn't change that much because we are indeed in the pattern and we have reached this point that it's near the first state. Okay, so we see that the overlap is close to 1. And in this case, the attractor is not switching. Okay, the dominant attractor of the network, it's itself the one that we wanted to go. Second, well, if we see in the no retrieval region, here we have an overlap that is less than the critical overlap and a mutual information that is less than the critical mutual information. So what we see here is that we have this network and a state there, and these patterns are not there anymore, so our state goes beyond these patterns. So in the dynamic, what we see indeed is that randomly throughout time, it changes the state of the network without reaching a certain pattern throughout time. We see the overlap that it goes nearer to 0 and it passes the overlap threshold that we have imposed. And we have an attractor that is all the time switching, yeah? Last, this state that we wanted to reach, which is the third one, in which we have an overlap that is greater than the critical overlap and a mutual information that is less than the critical mutual information. That's what we have imposed, and what we are eventually saying here is that we don't have a pattern. I mean, I'm going to show you then, but it's like a confusion state, okay? So if we have this neural network, we have a state there, it's like it goes to another one. So it's not to the closest that we should go. And the dynamics, we see that through time, we see that the network changes, but not so much as the other one. The overlap doesn't go to the pattern that we want to go, but we see that it actually switches to another pattern and it's stable through time. So this is basically the phase diagram that we have looked for, and to search for the critical lines that set the threshold, we assume that it would be 1 AC1 for the separation between healthy retrieval and pathological, and AC2 for the no retrieval and the pathological state.
So in the end, our conclusions are that the introduction of anti-habit learning destabilizes dominant patterns, preventing pathological fixation, while decay and synaptic loss affect memory retrieval in qualitatively different ways. Phase diagrams reveal well-defined information thresholds, separating retrieval confusion and non-retrieval regimes based on no overlap in mutual information measures. The proposed frameworks provides a minimal and interpretable model to study memory degradation, and memory loss arises from progressive destabilization of stored attractors rather than explicit deletion of stored patterns. So this is our research, and thank you all for listening. And a special thank you to Mauricio and Rocky, to LASCON and the Ibro Foundation.
Okay, very nice work. The stored patterns and are they very sparse, or they sparse, right? What's the sparsity? It looks like half of the net.
You mean, sorry, this one?
The patterns you store to create the attractors. How do you choose?
Oh,
these
they are
patterns?
randomly.
Right.
They are
And
randomly
half
chosen.
of the neurons are activated. Do you know the sparseness in this? So you
Sorry.
have 10 cells, are on average 5 of those 10 cells activated or 1?
Okay.
That's the sparsity.
Okay, so we kind of like set a random value for that as well. Yeah, because we don't know exactly how to measure that, but yeah, we just took random like set of states and also a random way to store those firing and not firing
Because
states.
it would be interesting as the field nets depends principally on the sparsity
Yeah,
of buttons
mhm.
and the overlap. If there's already overlap to random fluctuations, you lose capacity will be.
Okay.
What about, are there cues to retrieve patterns, or just it's a driven by random noise, or just Bayesian hopping?
Yeah, we just added this noise, and we just like, we search for an actual pattern to begin with, and then we like move it a little far away for us to know that we want this pattern to go to, you know, like, so this noise that we are adding to the model, it's that.
It's like a cue.
Yeah,
Right.
yeah. Mhm.
Nice work, thank you.
Thank you.
It's related to what Boris said. So you just use random patterns. It would be interesting if you introduce some correlation between the patterns, and then you could study your retrieval of the memory based on the correlation between them. And also, have you studied the dependence on your results on the size of the network, the number of neurons?
Yeah, totally. I mean, maybe I haven't mentioned it very clearly, but in this case, I mean, when we are seeing this phase diagram, we are actually doing an analysis based on the capacity, which is the patterns related to the quantity of neurons. And this is the learning gain that we are adding to the connectivity of each in between neurons. Okay, so for Hopfi model itself, we have a capacity, critical capacity, you know, but in this case, we kind of see, but because of the noise and everything that we are adding to the model, we see this behavior. Mm-hmm.
Any other question? No? Yeah, thank you again.
Thank
and
you.
Let's go for coffee break.
Hello? It's okay? So good morning, everyone. It's a pleasure to be part of LASCON. My name is Anna, and today we present the modeling seizure dynamic with epileptor. So first of all, it's important to understand what is epilepsy. Epilepsy is a neurological disorder characterized by a persistent predisposition to generate epileptic seizure with important biological, cognitive, and physical psychosocial consequences. So epilepsy affects 15 million people worldwide, and 1/3 of the patients develop drug resistance. So the seizure is abnormal, is a breath event that is abnormal, hypersynchronous, and that happen in the brain. So although seizures may appear unpredictable at the individual level, their probability is strongly influenced the global brain state and the sleep escape modulator. So in non-REM sleep, we have more synchronized than the REM sleep. And in this project, we want to understand this dynamic, and our hypothesis is that sleep states do not only change activity pattern, but also reshape brain dynamic, influence how close the system is to the seizure transition. So in non-REM sleep, we have a reduced dynamic safer monitoring, and it's closer to the seizure boundary. And in the REM sleep, we have more desynchronized and increase the stability and farther from seizure boundary. So in the first part of our project, we want to understand the coupling in the thalamocortical system, and we want to compare non-REM and REM sleep. And the second part, we want to see all the brain, use the virtual brain, the TVB. So instead of study seizure like an isolated event, we want to understand this dynamic, and we use the epileptor model that will allow us understand where's the seizure start and the sentinel node bifurcation, and we can see here the seizure, and here we can see the homoclinic bifurcation in the end of the seizure. And this is controlled by the slow variable.
So, good morning. My name is Arthur, and I'm going to distinguish this model architecture we created. So from here, we focus on the mechanisms linking sleep state to seizure susceptibility. We couple a 6-dimensional cortical epileptor to a thalamic Wilson-Covan mean field with a thalamical cortical population excitatory and reticular inhibitory population. So sleep state is implemented by acetylcholine and modulation, and in the non-REM sleep, thalamus tends to oscillate and cortex is set close to instability. And in REM, thalamus is more tonic and cortex it's more stable, like you see here. And the G variable here, it's the ultra slow variable on our equation. So the key pathway we analyze it is the thalamocortical to cortical X1 delivered through an AC coupled synaptic filter. So the cortex mainly receives thalamic fluctuations rather than tonic baseline. And now I will show how the epileptor produces fast seizure transitions. So we used this piecewise cubic function here This is the fast epileptor subsystem, where a seizure appear as rapid regime switches. And the core feature is this piecewise nonlinearity F1x1, which creates the geometry of beam stability and seizure-like excursions. Thalamus enter in this part like this ITH in this fast subsystem equation I've shown here. And the recovery variable follows this other equation below the equation of fast activity with thalamus input. So basically, thalamic input perturbs X1, while Z shifts the landscape slowly, changing how easy it is cross the separatrix of epileptor. Next, what controls this slow shift is Z and the parameter X0, like you see here, the introduction of this X0 parameter. So the slow variability, this slow subsystem, are governed by this Z variable, which acts like a permissivity or vulnerability state. And it involves as the key parameter, X0, which states the system's distance to instability. In the non-REM, X0 is set to make the cortex closer to transition, and in REM state, X0 pushes the cortex deeper into the stable basing. So even before thalamus, REM, it's modeled to produce spindle-like dynamics in the non-REM. This is the thalamic mean field model. This is the equations we used by thalamocortical population and reticular population with low acetylcholine and high acetylcholine by non-REM and REM. So Kovan model with TC and RE population using standard rate equations, and we used this sigmoid activation function. So by changing the neuromodulating settings, the thalamus switches the regime. By non-REM, low acetylcholine favor the oscillation activity consistent with spindle band rhythms, and REM with high acetylcholine stabilizes the atonic fixed point and reduces the rhythmic fluctuations. So sleep state changes the spectral content of thalamic input, but whether this affects the cortex depends on the couple filter. And this is the coupling filter. This coupling is designed to pass fluctuations and suppress tonic components. First, an adaptive high-pass stage computes, removing the baseline, then synaptic low-pass shapes, it producing the effective of thalamic input to cortical X1. And this meaning it's the non-REM oscillations survive the high pass and rich cortex, and the REM tonic activity is largely removed, so the cortex receives little effective drive. Now the results, phase space analysis. Here you can see the dynamic consequence in non-REM. The trajectory crosses the separatrix I mentioned before, and the trajectory and enters a seizure-like regime. Large excursions consist with ictal dynamics, and in REM, trajectories remain near the stable fixed point and return small perturbations. So this is the Bayesian geometry story we trying to send here. This is the 6D the dynamics projection in 2 dimensions. This PCA projection summarizes the high dimensional dynamics, interictal activities clusters in a stable region, while seizure trajectories form a distinct orbital-like path in the reduced space. Here you can see the higher Z and movement into a different region of this state space, and this supports the interpretation that the non-REM enables state transition and ictal manifold. So at final, we run Monte Carlo statistic. We test paired Monte Carlos across 30 seeds, comparing non-REN and REN using a same random initial conditions per pair. Seizure probably is very high in non-REN and near zero in REN in this configuration, and we paired with a McNerman test. So in conclusion, cortex X0 sets distance to transition, thalamus non-REN generates oscillation, REN it's tonic, coupling AC filter transmits oscillation, suppress tonic drive. So next, here we close this this hypothesis and we start to work with the epileptor in a whole brain model.
So this is where I take in. We use the TVB, virtual brain model. It creates a dynamical system for each brain area, and it couples these dynamic systems with based on the connectome, so structural connectivity. The equations are pretty much like the canonical epileptor. We change this coupling, right? And this X0, which is the excitability that works on our slow variable. So if you have one excitability lower than the critical value, which is minus 1.6, you don't have these seizure-like oscillations. If you're higher than minus 1.6, you're gonna have these oscillations. So here I set X0 higher than 1.6 for 3 subcortical areas on the left hemisphere, are these in red, and close to the critical value, minus 1.7. Here for these other 2 areas, also here in the cortical part of the left hemisphere. From this we generate a signal of our sources, which is each of these sources is one epileptor model with same parameters here in the gray lines and the different parameters, as I said, on the other color lines. To go from the source signal to the EG signal, we use this gain matrix, which it makes a linear combination of our source signals to compose this EG. So here we're not in the source space anymore, we're in the sensor space. These are our EG electrodes, and you can see there's a very large spread of the seizure activity through the whole brain. And sometimes it makes it difficult to visually detect where the seizure is coming from. So what I wanted to do was use a functional connectivity technique. My advisor talked about this the day before yesterday. We take our time series, or EG signal. We see what patterns a point makes with its 2 next neighbors. So if you have 3 points, you have 6 possible permutations. You have 6 symbols. Then you compare the sequences of symbols from 2 different parts, 2 different electrodes. We compare in a window of 20 samples with 90% threshold. So to have a match, if you have 18 out of 20 symbols matching, you're gonna have a connection on your connectivity graph. And this sample window travels through the whole signal, and we sum it all afterwards and normalize. Then we have this ponderated graph, weighted graph. And doing that on the TVB signal, so here we have much more activity on the left hemisphere, as we can see also on the adjacency matrix. And the edge weight distribution, so you're gonna have close to 3,000 edges here. Only a few of those are in this high connectivity range. And they're all mostly on the left hemisphere, so where we are having activity that generates the seizure. And finally, I want to show that this is present even when the seizure hasn't spread throughout the whole brain. So this is just 1 second sample. This is just 1 second of our source signal and our EEG signal. And this is the corresponding connectivity matrix. So for this 1 second, we can see there's a laterality, which we can't see just by looking at the signal. So that's it. Thank you very much.
So if I understand correctly, you put the epileptor everywhere in the brain. Is there a configuration where you would put the epileptor only in the focus, the epileptogenic zone, and have the rest of the brain using other neural mass?
So this is what we tried on our simplified model. We use a Wilson-Cohen for the other areas and an epileptor for the cortex. It's hard coupling and it's hard to parameterize. So the TVB uses epileptor for the whole brain, and it simulates where you don't have a seizure activity by using that excitability variable. Because when it's very low, you're gonna have oscillations that don't look like seizures. They can fool you. They look like normal neural activity, but when you have a higher excitability, that's when you start having seizures.
You can put this higher excitability only locally. You're not obliged to do it uniform.
Yeah.
That's what you say.
I choose only a few areas to put a high excitability, and the other ones are low excitability, and those are coupled. So the activity spreads even when you have low excitability. And that's the reason why we've used epileptors.
And another question is that we know that different seizures are related to sleep and others are not. Like in the sense that may display the intellectual spikes, for example, for some seizure types, and others, the sleep is completely devoid of any epileptic phenomena. It's like a pause during sleep. Did you see anything like this by playing with the parameters?
Yeah, we see that in parameters, like some of our results are not so good with other parameters, and like we using this Wilson-Cohen model because when we try to use this neuromodulation biological in thalamus with Hopfield or other models, we have a problem with scale by a mean field and micro model, and we try different parameters and different models to do these results. So yes, you are absolutely right.
So in your abstract, you mentioned this dynamical safety margin, which is a concept, of course. In your model, you just have non-random brain sleep. How does this safety margin would depend on the parameters? I mean, do you test? I mean, which parameters would have a largest effect on this?
The X0 and the Z have the more effective on our parameter. So it's very clearly when you put these parameters much higher or low, you approximate it or not of the separatrix.
Just to find out, how much of this work you did here, and how much did you work you did before coming to LASCON? So everything was done here.
Yeah, I work with non-Hermitian complex systems, not epilepsy.
Okay, thank you. So we come to the next.
But it's okay there? Yeah. How to Okay. Present acá. No veo dónde. ¿Eso? No. Okay, so hi, we are Lucila, David, and Mateo. We are going to present our project. So we want to explore the hidden Markov models for modeling stochastic voltage-dependent activity of ion channels and the influence of noise in the single-channel recordings. So this all started because back in Uruguay, in my lab, I don't work with this, but some colleagues do. They do patch clamp experiments, and they actually do single-channel experiments to have records of a single channel activity. Is a good way to characterize an ion channel when you don't know much about it. And you do it with patch clamp, patching a cell, but you actually need to have this kind of configuration when you have only one channel on the record. So you, for example, can change the voltage of the membrane, and you can see how this channel behaves, okay? So this is the cell for when they do it. But they pass me this data, and we say, okay, it would be amazing to make a model of this channel. We start to work with this. We present the project with this idea, but we have a problem. We can't use this channel, okay? We have like a complex behavior. Then we ask, what about that to the people from Uruguay? And they say, oh, I send you the whole cell recordings that are not outside out. I mean, you don't have only one channel, you have who knows what. So now it's a problem. So we need to call los simuladores, okay? We need to simulate a channel to be able to continue with our workflow and with our idea. So what we do, we take another channel, an already simulated channel from a paper, that one, that was on ModelDB, and we use this channel that already has a stochastic transition between states with a defined topology, and we say, okay, let's try to replicate this channel. Let's try to model this channel and try to see what is the method, okay? So one first thing that we do, that later you're going to understand why, we change a little bit the channel. This was the original topology. We change it to this one. We change like the conductance of this state to be an open state, and we also like adjust a little bit the kinetics to don't have a channel that opens all the time a lot. Okay, so now we have like our own channel, our ground truth. The slice is, oh, well, okay. So we have this channel, we have these kinetics of the channel. I'm sorry for the layout of the slice, it's changed. And we have this channel on Neuron, okay? We can import it to Neuron, we can put it on a Soma, whatever, and we can study. What do we do on Neuron? Okay, we perform a voltage clamp protocol. If you want to understand the voltage dependency of a channel and how it opens in different voltages, you want to do this kind of experiment. That is a very common experiment on a setup of electrophysiology. So we put the membrane at different voltages, and you can get the membrane to be in a steady way there, okay? So it doesn't change. You have the current going out or in for the channel activity, but the voltage stays the same, okay? This is like the layout of the simulation on Neuron, and you are going to end up with recordings of the channel at different voltages. We did to all these voltages, just presenting here 3. As you can see, the current is going outside because it's a potassium channel, and it's once you increase the driving force of this current, you're going to have like bigger current, okay? And you also can see that we have only one channel because we can decide that it's Neuron. So we put only one channel with stochastic activity, and you see that you have or it's open or it's closed. Okay, so at first opens a little bit, then it starts to open more, and then opens a lot more, okay? So this is like the voltage dependency of the original channel. What we want to do, oh, sorry, we don't have noise, okay? It's a perfect register. You're never going to get something like that on an electrophysiology setup, so we're going to try to work on that. And, okay, let's do some analysis of these records, and let's try to understand how this dynamic, how this voltage dependence activity can be modeled, okay?
Okay, so to get the hidden Markov model for this channel, we use an approach described by these authors of this paper, that they used a deep learning approach in order to do this. You can do it in other ways, but we wanted to explore this more sophisticated or new method of getting the hidden Markov model. So with the recorded data that we have like here, in this case we have only the idealized time series because we don't have the noise. You have an open and a closed current. And with defining the threshold, you can get the 2D histogram that it contains all the information about the kinetics and the transitions between the two currents. And you feed that histogram to two neural networks, convolutional neural networks. You fed these networks with this image, and you will get one vector for the probability of each possible topology, because you have 5 states. This method is only implemented for topologies of 5 states and linear. And to the other network to get the 8 transitions. So you get the values of each of the transitions between the states. And as these networks are really hard to train and it takes a lot of time and resources, we use the available networks that the authors uploaded. There were 12 trained neural networks for the topology and 2 for the specific topology that we had, that it was close, 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 open, open. That's why we changed the model of the channel in the first place, in order to be able to use this train network instead of having to train one from scratch. So first, for the topology classification, you have the different recordings at each voltage. You will feed this information to the networks separately. You don't, like the voltage dependency, you only do it separately. That's a problem that we will discuss later on. But for each voltage, you construct the histogram and then feed it to each of the 12 neural networks that we had, and we get the vectors of the probability for each topology. Then we decided to get only the probabilities for the networks that were like certain about the decision. So the probabilities that were below 0.5 for any of the topologies, we discard them. And then we did a voting rule and decided the topology for each voltage based on the majority voting across all the neural networks. So this was the possible topology, closed or open, for each of the 5 states, and we knew that this one was our own topology. So then we analyzed the performance of the neural networks in order to get the topology, and we can see here the number of votes that the right topology got for each of the voltages, and we can see that in the range in the middle, that it's like the more rich range, because we have like the most transitions of being close or open and not very close or very open like in the extremes. We get a very good performance and effectively decides that the topology is the right one. And when the voltage is lower, you get a topology that is most close than the right one, and in the other extreme, more open. And then for the rate of estimation, we used one of the 2 neural networks that were available for this topology, and we feed the 2D histogram again for each voltage separately, and we get the 8 transitions between the states. And we did this for each of the voltages that we had. Oh, this is okay. And we get these transitions that are not very similar to the ones that we had in the first place. And this is a problem also related to the fact that these are not continuous, because we're doing this separately for each voltage instead of feeding like the whole set of recordings at once. But okay, we can, we were guessing, okay, how can we evaluate the performance of these transitions, right? Like the performance of the new network to get these transitions. So we could compare the real transitions with the predicted ones, because we had those, because it was a model. And here we have the results. But the problem is that there are many models that can get you the same kinetics. So instead of comparing the parameters, we can compare the performance or the activity that these 2 models give. So we repeated the protocol for the voltage clamp at the same voltages, and we got again each recording for each voltage, and then compare the probability of finding the channel open and compare the 2D histograms that are fed into a network between the simulated and the real model. And we can see here that again, for the regions in which we had the right topology for the neural network, we get a small error. Here is like the 0, and this is plus and minus 20% of error. And here, this is the distance between the two histograms, and we see that the smallest error is in the range in which we had the right topology, and we have a higher probability of finding the channel open in the lower region, and a lower probability of finding the channel open in the higher region. And this is related to the fact that these are discrete and not a continuous, and the topology wasn't correctly in these two extremes.
Now talk about the noise. We add the noise in the signal for two main reasons. The first, because the signal obtained for the simulation is clean signal, and you know that a real signal is very different from that. And the second, the paper mentioned that his model is robust for the noise. For the reasons, we decided to validate the model under three different scenarios: modeling with no noise, modeling with low noise, and modeling with high noise. Okay. Here we have the signal with no noise, and the other types of noise we applied in the signal for obtain the final signal. The first type of noise is Gaussian noise or thermal noise, is a noise of the high frequency. The second, pink noise, is the same frequencies high, but is a flicker noise. And the last is drift noise, is for the variation, the baseline frequencies of the signal. We have the final signal with the conjunct of the signals. Okay. In here, for make analysis to the model, we also make a comparison with the intensity of the signal. And here in left, we have the signal with low level noise intensity. In right, we have a high level noise intensity. For the comparison, after they apply the noise in the signal, we repeat the process that Luli mentioned before, but that's only difference. Apply the high pass filter for for the model. High frequencies is most important, and for this apply the high pass filter. And this is results of obtained with the model. It showed good efficiency in the classification, the topology in the three scenarios. The model correctly identify the topology in all scenarios and in same ranges. And when it made mixtape, shows errors happened at the similar voltage range as well. This same behavior is observed in the other metrics, such as the open probability and the 2D histogram at a distance. And so well, you can see that the model is efficient both for a complex clean signal and for signals with a high level of noise.
So in summary, we are ending our presentation. So the good, the bad, and the ugly is a bad. Okay. So we believe that we can say for the good things that, for example, Neuron has an already good implemented tool for doing Markov modeling channels. So if you want to do that, you don't need to implement nothing new because it's very good for that. We don't show this, but we validated that you can like extract the same values if you just put again the kinetics. So it's a good thing. Also, another good thing is that we believe we have a good fit from a certain range of voltages. Not for all of them. We have problems on the extremes. Luli is going to talk about that. But on the more rich range of voltages, when you have more transition between states, we believe it's a good fit. And also, okay, we developed like an automated pipeline to do this voltage-dependent iteration. To understand the voltage dependency of the channel, we need to do an iteration between all the recordings. So that's also a good thing, and we actually learn a lot. So I think that's the most important part.
Then for the bad part, the problem is, well, first that the only topology that this approach can be used to is the linear 5-states topology, and that the neural networks that you need to train take a lot of time. And the other problem is that this voltage dependency cannot be modeled as a whole thing altogether, and you have to do it separately. So changing the approach for a joint fit receiving a set of multiple 2D histograms would be better in order to have a smooth transition between the voltages. And yeah, well, this gives rise to this reduced performance at the extremes of the voltage range.
So the ugly is this plot, awful. And this is our pipeline. We want to thank all the lecturers and Rothbard supporting us a lot. This is the references. This is us. And thank you very much. We have all on GitHub if you want to look. So any questions that you want to ask us? Very welcome.
Thank you. So if you wanted to use a different kinetic scheme or what you call topology, you would have to retrain the net, but it would work for any.
Yeah, they claim that. We actually get in contact with the people who wrote this paper because we asked them, okay, maybe you have like a trained network for our original topology and you don't publish it, but maybe you can send that to us. They say they don't, but they are like working on a new model that is not so restricted with the linear thing. And also they talk us about they are wanting to implement this voltage dependency kind of analysis to be able to put a lot of records and not only one by one. So they are working on that, but they weren't able to share any of that with us. But they were very kind.
Going back to the bad part that you mentioned, how you imagine that this joint fit across voltages that you mentioned that would improve your performance?
Yeah, the thing is that we're forcing the model to have a topology in the extremes that the activity is not fitting into that kind of behavior. So first you decide the topology and then you decide the rates based on the topology. Like you choose a neural network that is trained with data from that topology. And the problem is that as you're not using everything all together, it's not fitting correctly. Like the rates are not fitting to the topology that you are trying the model to fit in the extremes. So if you do something all together, the rates would adjust to fit into the topology that you are deciding for the whole thing. Not like a discrete thing.
Neuron, these transitions are modeled by an equation with a curve, and we try to fit, but in the way that the network guessed the kinetics, it was impossible. So maybe an implementation that you can consider, okay, I know or at least I try to guess that my voltage dependency follows this equation, this curve. I think it will be a very different outcome, yeah.
Thank you. Yeah. Next group. Arndt, I'm sorry, we don't have time for them to answer your question, but I mean, they can read it, because Arndt sent a question to the previous group. They can read it and then they may be able to.
So hello, good morning everyone. My name is Gabrielle. I will present the project that I work with my colleagues Federico and Lindsay. I would like to start, thank you all for the opportunity to present here at LASCON. So our project was to extending a computational model of behavioral timescale cymatic plasticity, also called BTSP. All right, so first I would like to introduce what is the BTSP. It was presented in this paper in 2017. And if you had already, probably yes, like learned a course in Introduction of Neuroscience, you're already familiar with the classic Hebb's law that says that neurons that fire together wire together. And for this law, it's important that a neuron fire right after a presynaptic neuron in only a tenth of milliseconds scale of time. And it has to happen like many times with repetition for this rule of plasticity. But the BTSP rule for learning present a different dynamics for a kind of learning that happens over many seconds and with no many repetitions. And is also founded in the hippocampus area CA3 and CA1. And to understand these BTSP, it's important to know what is a place cell and what is a plateau potential. So place cells can be found in the CA3 and CA1 area. Those are cells that, let's say, if in an experiment there is animal in an arena and it fires in a specific place of the arena. I'm sorry, I need the water. Okay, and a plateau potential was presented by this paper in 2015. That was very important for the BTSP paper. It happens when a neuron cell from CA1 area receives an input from a place cell in the CA3 and from the entorhinal cortex. It has to receive this input at a very similar time. And what happens is that it changes the membrane potential from the cell into CA1. So here in the first graph, there is resting potential. And when it receives the input from CA3 and EC, it creates a despolarization, a long-lasting despolarization that is called a plateau potential. And after, if you repeat these same experiments in the same place, this plateau potential will become a burst of spike that is called a place field. So in the BTSP paper, the experiment made was a mouse in a field, one-dimensional field with different features. They were recording one neuron cell in the CA1 area. And then they ran this experiment many times. And on the first 9 laps, they didn't saw any changes in the membrane potential or the frequency. And from the 10th lap, so they could see the calcium plateau. And then after they repeat this experiment, they saw this burst spike happen again. So here is the calcium plateau. And on the figure B, there is the graph of the membrane potential on the lap 10s. And then on the lap 11s, it shows the plateau potential. And after, on the lap 27th, in the same place, there is place field that they describe with this burst of spikes. So the paper we used to extend was this behavioral timescale plasticity of place fields. It was a mathematical analysis. from Shuval that we call, it's called Shuval model. So this model, it presumes that a mouse is in a track of one dimension with features inside it. And it's recording inputs from a population of CA3 cells to one neuron cell in the area CA1. And those inputs from CA3 are described as simple Gaussians, and it is a firing rate model. So those are like numbers that this model use as input for CA1.
Okay, so I'm going to go pretty briefly through the math of the model. I'll try to pass this real quick. So as Gabriel said, this is a firing rate model. So the firing or the memory potential of the CA1 cell is modeled as a linear combination of the inputs of these CA3 place cells. And this is a plasticity model, so we have to show how the synapses changes, and this is following this differential equation, which is a linear combination of a potentiation trace and a depression trace. And both are modulated by this P, which represents the instructive signal that we said comes from the entorhinal cortex. And these terms W and 1 minus W are there to represent the fact that resources in a synapse are conserved. So you have, for example, receptors that are exo or endocytosed, but they are conserved, the total. And then this is how the traces evolve. So in the model, both traces, LTP and LTT, potentiation and depression, they evolve through this equation, and we use Euler method, and Shuval also uses Euler method. And then the instructive signal from entorhinal cortex is modeled as a Gaussian pulse, as a pulse that starts at some time random, and then it decays exponentially. And this is how the traces evolve in time according to Shuval's paper, and this is the place cell they get in CA1. So what we wanted to do in our work is to extend this model to become a little bit more realistic. So in the original paper, they have constant velocities, which is something we know animals usually don't do. And also they only model only one place cell in CA1, and we wanted to check if we can get some more realistic statistics of place cells, and you can do statistics with only one place cell, so we did many. And it only simulates one-dimensional trajectories, so we also extended to 2 dimensions. And about these statistics, well, there are these papers that say that the peak firing rates in CA1 should be log normally distributed, that the sizes of the place fields should be also log normal. And also there's this work from Tani that shows that place fields number and size correlate with proximity to walls. So you have many small place cells close to walls and fewer and bigger place cells in the center. And then there's this paper also from Sosa et al., but there are many that show that when you have an animal that already knows a context and you then repeatedly put a reward in a place, then place fields changes and you have more representation of the position where the reward is presented. Okay, so these are the inputs we generated. We simulate 2-dimensional trajectories of an animal walking for around 6 minutes in a square arena. We change the velocities, so now they are normally distributed instead of a constant velocity, and we simulate Gaussians in 2 dimensions that represent the cells from CA3. Okay, so this is basically what we got. This was actually pretty easy from the model from Shuval. In less than a week, we already have the model for what we wanted working, and this is the place cells that are generated in CA1. And yes, after a lot of trying with parameters, we got the statistics we want. That was not that easy, but we got, this is the firing rate, peak firing rate distribution, and we see that this is the logarithm, and it pretty much resembles a normal distribution, and actually this is p-value approved. So this is we can't discard that this is a log normal distribution. The same with the area of the place fields. And initially we didn't have the position distribution that we wanted with more place cells close to the walls, but from looking at the model, we realized that the position that the place cells represent only depends on when you get where the simulation is when you reset the plateau potential. So instead of using a uniform distribution, we define the distribution that is bigger close to walls and smaller when you are in the center, and that obviously get us the result we want. But we had to put that by hand. And the same happened with the sizes of place fields. It worked with that. So that was very happy, and it was only the first week, and we were very happy, but we came up with this paper which says that actually place cells are formed first in CA1 and then in CA3. So main hypothesis of our model is that we learn place cells from place cells. That doesn't work, so we had to come up with something else. Luckily, in a talk from this week, I think, Alessandro Treves mentioned this paper, which they basically show that you can get place cells with the statistics of place cells only by having a Gaussian process. So that's like a normal distribution data that is smooth in space. And you threshold that, and you can get the statistic of place cells. We did that by doing linear combinations of exponentials and choosing the weights and the centers and the amplitudes randomly. And we get inputs like this. So this would represent combinations from, I don't know, grid cells, from terminal cortex, place cells from CA1 that are from CA3 that are still not formed, and stuff like that. And then we had to slightly change the plasticity rule we had. So instead of having what we had before, we throw the LTD term and we replace it by a term that normalizes all inputs that come from that come to one cell. So if one cell has a lot of has very potentiated input from one cell, then it has to depress the connections to other cells. And again, we got place cells after a lot of playing, and we eventually get the statistics we want as well. We have to say though that this isn't exactly stable. So if you run the model many times, you don't always get the statistics perfectly. This is probably due to the fact that our model inputs are not totally random. Weights are only positive, and the learning rule imposes some non-randomness in the input. So the Gaussian process is not exactly Gaussian from the inputs.
So then we asked about how rewards influence the place field formation. So you see in the first one, I guess, they're around the edges, and then they cluster around the rewarded locations, which emerges directly from the BTSP rule. We also tried remapping, so we tested the model across different contexts, and when the context changes, the spatial representation also reorganizes, which is consistent with behavior of hippocampal place cells. And then we also looked at the gradual changes in the representation as the environment changes smoothly. So it shows this continuous, I guess, transition, which is also consistent with how place cells behave. So in conclusion, we extended the model for variable speeds for multiple CA1 cells, 2D trajectories, non-place cell inputs, and it's consistent with statistics and remapping and morphing that is associated with the actual behavior of place cells, more experimentally accurate. However, of course it has limitations. There are too many parameters in this model, which makes it very difficult to augment. We also have a problem with repeatability, as Fede said, and the instructive signals are all imposed manually, which is also a problem. So this is a start to a more experimentally realistic model, but those are things that could definitely be improved. So, yeah, thank you.
Questions?
If I understand correctly, the modification that you had to make is a sort of homeostasis rule, right? Which would predict that you don't need the LTD, but the synapse somehow renormalize. There is an experimental data that are in favor of that. I don't know if you know the paper, but I don't know for the hippocampus, but I know for cerebral cortex that it does happen. For your information. Maybe because I'm not a do not know so much well the hippocampus, but in cerebral cortex it could be a source of inspiration also for your model.
Very nice. So how much of this project was actually developed here? Do you work with all of it? Okay.
So yeah, I do work with place cells, but not with BTSP. And yeah, we coded everything from zero. Well, the model from Chuval was in ModelDB, so we started from there and we updated that.
It's called Chuval, not Federico.
Yeah, that's why it doesn't have our names. Yeah. Thank you.
So do you have any idea of the next steps to try to improve it? I mean,
I think the best, or the next step that would make it easier to improve it further, so that the most intuitive next step would be to give it less parameters. Right now there are so many parameters that when you test the model, we had to go through
but how
quite a
could
bit
you
of
reduce the number of parameters? How could they constrain them?
I mean, we would probably need a cluster to do some sort of a grid search and go over different parameters and different parameter combinations in order to find, but it is a long way to find that. I don't know exactly how we could do it just to simplify it and remove parameters entirely. I think that the best way would find parameters that are consistently perform well in multiple different environments and then use those. So.
Yeah, and also one way of reducing parameters is the fact that there's actually degeneracy in parameters. We have many stuff that could be simplified right now, and because of the time we had, we didn't want to try that, but there's probably degeneracy. And one other thing that could be improved is what we were saying here, the fact that right now, when and where place cells are formed depends only on this instructive signal that we choose. So if we want to represent a reward, we say, okay, our reward will be here, we have more plateau potentials. But this could be something that could come up from the model itself. So for example, you could have like a prediction error that says, I don't know this position very much, I want to learn this more, I generate more plateau potentials, and something like that. That would be interesting.
More questions? If not, thank you again and have an interval now. Thank you.
So we are Natalia, Sabrina, and Claudia, and we are going to present a biophysical modeling study of cortical stimulation. So we are going to start to introducing you the Blue Brain project, which is the basis for what we have done. And then we will also talk about Avera et al. Model, which is, well, the neurons that we are using come from that model, our objective, methodology, results, and some conclusions.
So, the Blue Brain project, these people modeled the rat brain, and they found more than 30,000 cells and more than 37 million synapses and more than 200 cell types. As you can see here, the diversity of morphology types of the cortical layers, including pyramidals, neuroglial cells, and the axons in blue and the dendrite in red. And they also checked the electrophysiology of each cell type, and they observed a firing pattern. As you can see, delayed, continuous, bursting. As you can see in this figure from his paper, it shows the diversity of firing patterns in the neuron connections. And Avera et al. 3 years later, based on the Blue Brain project, and this work shows 5 kinds of cells representing the 5 layers of cortex, and they simulated the cortical stimulation, and they used intracortical microstimulation and uniform E field to see how these cells behave in presence of these stimulations. And with this subset of representative cell types, they chose 5 cell types, and they created 5 clones of each one, 25 in total, and they stimulated all of them with these two kinds of stimulation, and they concluded that the effects of the stimulation depend strongly on the axonal morphology. That's why we are also always emphasizing the morphology to see how the electricity behaves.
Therefore, the principal adaptation of Avera from BBP model regarding with cell types is the broad excitatory inhibitory diversity across cortical layers, while in the Avera adaptation used a subset of representative cell types with 5 morphological clones per type. In the cell model, BBP used a multi-compartmental conductance bus, and the Avera adaptation used the same model but adapted for stimulation response studies. And one of the most important difference between these works is regarding to the morphology. And the BVP used a realistic morphologies, often from juvenile rats, and use unmyelinated axons, while in the Avera adaptation, the BVP morphologies scaled to match adult rats and adult human geometry. And there was a strong focus on axon features like diameter, orientation, terminal branching, and myelination. And the principal goal of BBP was biological realism for neuron circuit behavior. And in the adaptation was a response under different cortical stimulations methods. And based in all these adaptations, the authors confirmed that the effects of simulation depend strongly on axonal morphology. So our objective is reproduce a minimal subset of the results reported by Averedo 2018 by rerunning a simulation of cortical stimulation current on selected cortical neurons models ported from neuron to the net-by-net frame. To achieve the same, we use a program language Python, a simulation engine neuron, and modeling framework net-by-net. We choose three types of cells from 3 layers. The layer 1, the neurogliaform cells, an inhibitory cell, and the layer 2, 3, and layer 5, both pyramidal cells and both excitatory cells. We simulate an input current extracellular to approximate the response of the effects of the electric field using this stimulate current range in these cells layers. Within each layer, the simulation amplitudes were set based on the cell types with the lowest extracellular activation threshold.
So for our results, we first visualize the morphology of each of the cells that we chose. Here we show one from layer 1. Layer 1 is characterized because it has very few somata, and also the principal cells in this layer are neurogliaform. So these are circular shape neurons with very small dendrites, and they have a thick and dense axon. Then for layer 2, 3, and 5, we show pyramidal cells. These cells have base dendrites, and they have also apical dendrites. And the main difference between these two type of cells is that the ones in layer 2, 3, they present cortical communication, whereas L5, they go farther away to deeper subcortical areas. Here we also see the membrane voltage of the neurons, and we can see that it changes across the different types of cells and also across layers. Okay, then we also looked at the electrophysiological intrinsic behavior of these cells. And for layer 1, for neurogliaform cells, these present a burst non-accommodating pattern that refers that it bursts in the beginning and then the spikes remain. And the pyramidal cells from layer 2, 3, and layer 5, they present a continuous adapting pattern, which refers for a more stable spiking. We also, well, here we show a raster plot for the 15 clones that we have. The blue spikes are those from layer 1. Then we have in yellow those from layer 2 and 3. And in green, those for layer 5. within 800 milliseconds of simulation time. On the other side, we can see the population firing rates of the cells that we chose, one for each layer. And we plotted here the firing rates for different currents. So for a very low and negative current, they didn't spike at all. And then the cell from layer 1 and from layer 5, they have a similar pattern. They have high firing rate in the beginning, and then they show this more stable pattern for the simulation time. On the other hand, the cell from layer 2-3, in the beginning it shows also high firing rate, and then it decays, but then it presents these windows of spiking, and then a window of silence, and spiking and silence.
So now we simulated the stimulation. We tried to simulate the stimulation using the LFP. We stimulated the cells, one single cell, as the same way of the paper, and we put the electrodes in the soma, dendrite, and axon. For this layer 1, we see a high intensity in soma, and we can observe a silence in dendrite. And maybe because the dendrite is small, we have a weak dipole, and in the axons, the activity is moderate. For the 2-3 layer of pyramidal cells, we have high activity in the beginning, and this is soma, and it is according to its firing pattern, and that we see before. And we have a weak activity in dendrite, and a small one, a weak in the axon. It's important to mention, I will back here, I remember now, that in the code, the original mode is in neuron, and we adapted them, the code to work in NetBine. And to run the simulations, we have to do an adaptation to substitute the code of axon to a light axon in order to finish the simulation. We tried to use the original one, and it does not finish, and we couldn't present. And we followed an advice from, I think it was Fernando Jorge, that was here in the LASCON, and he suggested us to do this, and that worked very well. So let's continue. The layer 5, the latest one, we have a pyramidal axon with a large amplitude, and we have here the same continuous pattern in soma, high activity, and we have some activity in dendrite, and a little weak activity in axon, maybe due to it's almost in the end. So now we go to summary of our presentation.
Well, to summarize our results, we simulate three types of cells, a total of 15 cells, 5 per type, different with the adult, which use 25 cells, and input and simulate that applied the input current to see the change in the response of patterns. To conclude, we adapted the simulation from Aberrado 2018, originally implemented in Neuron, for use within the NetBine framework. We were able to we were able to reproduce the responses of 15 cells, 5 per cell, to a current injection stimulus. And we also conclude the effects of stimulation depend strongly on the cell morphology. We would like thank to 10th edition of LASCON, Principal and Professor Antonio Roque for organization and attention along the school and the opportunity to participate. Also, we thank to Valerie and Fernando for the supporting in developing this project. Thanks for your attention.
Questions? It would be nice if you could place your results together with the results from the original paper so that we could compare how well you reproduce the results, because only showing your results doesn't really tell us. I mean, you just say that you reproduce it, right? So just a suggestion for you. But I think the most important is how much of this you learned here. Did you know Neuron or NetMind before coming? No? Okay, so you learned everything and you did everything, LFPs, everything was done based on things that you learned here. The question?
But only something we tried to do that. This is the sorry. For example, this is our simulation, and this is the results of the paper in the shape. This is the simulation that we obtained with NetByNet, and this is the from the paper. So it's very similar.
It was not all because they didn't use the LFP to observe the stimulation, and we think it is interesting for cortical layers, and they reproduced the firing rate, but it was just one neuron. It's important to mention that we tried to work with populations. They didn't work with populations. And we would like to put the layers together to connect them. And we discovered that in the end of the last year, the Albera and his friends published one work connecting the layers. It would be difficult for us to do this.
So my question is, it was actually something related to this, is that you're showing the effect of the electrical stimulation on single cells, but we know that cerebral cortex is highly interconnected, and what is and it will cause, I suppose, some cascade of effects in the network that will completely change the activity and so on. Is this something that is easy to evaluate using modeling?
I would like to continue the work. I didn't know nothing about these tools, and never deeper understand the cortical cells. And I think it's very interesting to have many things to explore in this way. And we found these authors, and also the Valerie and Fernando Jorge that is in this equip, and some professors that passed here, and I think it's a great area. I like it very much.
So, hi everyone, thank you for your attention. I'm Giovanna, she's Amanda, and he's Lucas, and today we're going to be presenting our project to LASCON, which was inspired by Litton's lecture. So our project is a replication of CA3 microcircuit, which is a microcircuit of hippocampus, and this is related to schizophrenia. So, just to give a brief biological context, this is a really simplification of the biology behind it, but we have in this project hyperfunction in NMDA receptors within GABAergic interneurons, and these affect the balance of excitation and inhibition in the circuit, and because of that the gamma oscillations and theta oscillations lost their typical rhythm. So actually I will explain it later, but they need to be coupled to work typically, and because of that you have the outcome clinical output of that. The clinical output of that is cognitive discoordination within schizophrenia, like memory deficits, confusion, and other type of cognitive symptoms. It's coded here, but okay. The main reference for our project is this paper from 2020, which is Sheriff et al. And Litton is here. So this is the main circuit. It's kind of a complex microcircuit, which is the CA3 microcircuit. And you can see that we have 3 different types of cells. So pyramidal cells, which in this model is compartmented in 5 different compartments. We have PV basket cells, which are inhibitory, and also OLM cells, which is inhibitory too. We have different types of receptors that Amanda will explain it better later. And we have a lot of things going on here that we won't have time to explain it, but if you have any question, then ask later. And the most important things within this microcircuit is the ping-ing mechanism that creates the gamma oscillation, and the input from septal-medium region, which is related to the inhibition of OLM cells and the dendrites of pyramidal cells, and creates, let's say, theta oscillation. Okay, so this is really simplified. It's actually complexer than this, more complex than this. So here we can see a brain without schizophrenia. So what we want to see in the brain is this synchronization of the 3 types of cells. So this is the pyramidal one, and these other 2 are inhibitory, and they, as I said, because of their mechanisms, we have this pattern. So we have the theta waves, and the gamma waves are this little oscillation that happens here, and health cognition requires gamma bursts to occur within specific phases of the theta waves. So if it happens in another phase of theta waves, then you might have a decoupling of these waves, and then you might have the cognitive symptoms that I already said. And this impact is the information flow in the brain, which Lucas is explaining now.
Well, for understanding how this impact on NMDA receptors affect cognition in general, we will use metrics to measure information flow and synchrony. For measuring information flow, we will use normalized transfer entropy. There is basically, given that you know the past of some inputs, you have some uncertainty about its future, and you want to know how much this uncertainty is reduced if you know also the past of some input or some process that influences this other process. And in our case, our input will be an input to the pyramidal cells that is in neuron. It's implemented through the function netStim. It's kind of the middle term between a pure random Poisson process and a rhythmic process. And the output will be the firing rates or binary firing of the pyramidal cells. And to measure synchrony, we use this modulation index that is basically measuring this that Giovanna talks. That's for that gamma spikes should occur at the peak of theta waves and not uniformly across the phases. So again, well, this sheriff article is focused on pharmacotherapy. So we have these low values of modulation of synchrony and information flow in schizophrenic regime. And the idea is that since the NMDR receptor is with hypofunction, we can give some drug to scale its conductance. And the idea is that at some point this drug can restore or at least increase the information flow and the coupling between the waves. And they found this inverted U shape, that is, so if you administer some drug, it gets better, but if you give too much, it's not good. So our objective is basically to replicate this circuit in NEST and see if we, as using point neurons, will not have significantly changes that will mean that the main mechanisms between these was the dynamics of the network, the connectivity and the frequencies, and not the morphology of the neurons. And is this. So now Amanda will talk more about the methodology.
So good morning, guys. About the methodology, a network is basically neuron populations. Here we have the excitatory, that's pyramidal cell, and it's like 5 compartment cell. So this is important. Later I'll talk about a little bit more. And the PV cells are it's excitatory, and those two, PV and OM, are inhibitory. So the cells connect with each other, right, with a convergence ratio. That's the numbers here that you see. So for example, this number 20 means that for each pyramidal cell, 20 OLM connect to them. So, and also we have weights conductance for each connection, okay? And there's not here the numbers. And we have different receptors. So this is going to be important for our network dynamics. AMPA and NMDA have 2 time constants because it's a double exponential model. And the GABA has different values for each cell and connection. And okay, so beyond that, we have inputs. So we have the medial septum that is Giovanna told us about, that is inhibiting OLM and PV cells at a frequency of 6.7 hertz. And we also have the driving input that is stimulating the pyramidal cells in the soma and dendrites. And we also have a background activity that is this table here. And as you can see, we have a lot of connotants and constants and a type of receptors, different type of receptors, and we like use the same values in our network. And this is a low amplitude Poisson that we use in here. Okay. So our mission is to translate our model to a NEST model, and we use the HT neuron because it has characteristics that could let us work with that. So we have receptors, AMPA and MDA, GABA-A, and they have like double exponential, so they have beta function, which is okay for us to use it. And to mimetize the length of the fiber, we have a stimulant in different parts of our fiber compartment model. We use delays. So we estimated those delays using fiber length and cell disposition that we had in the original models. Just look at the neuron code and try to replicate it approximately. And we have in our model the rise of 2 important oscillations, the theta and gamma oscillations. So we needed to, the medial septum we detected, but the gamma oscillations arise from ping and ing mechanisms. That is basically a balance in the excitatory and inhibitory mechanisms. And we have also an important thing because we see that our population just got synchronized if we increased the delay of the inhibition. So we look in, there is nothing in the paper talking about it, but to model in NEST, we had to look for this these other literatures that talked us about the fast excitation and delay inhibition. So this was very important for our network to synchronize. And now, what we had to do is to change the parameters a little bit, because we have different model of neuron. So probably it's not the same parameters. We're not going to use the same parameters. So the contents of, like, we have here the ping and ing mechanism. So oh, sorry, there's no E here. The ing mechanism is basically generated by the external input in the PV cells, and the PV cell just inhibit itself. So here we have a large contents, 4.5, but in the ping mechanism we have excitation from pyramidal to PV and inhibition from PV to pyramidal, and we have very low numbers. So like we arbitrarily increase those numbers, and magically, like, the gamma oscillation appeared. Like we spent a lot of time trying to, oh my god, it's not working. We increased the delay, we did everything, and it was not synchronizing. And like, I don't want to tell you when, but we tried this and it worked. So we increased like very radically, like a lot. So the tools that we use in metrics was basically the same as the paper, just replicated. So the simulation, it was for 3 seconds. We excluded the first 2 seconds as the paper. We use raster plots, local field potential, PST, MI, and NTA. And we did the NMDAR scaling the same way as the paper. And the only difference that the LFP we used was a weight sum of currents in the excitatory cells, because we have soma and dendrites. We like did a weight based on the distances, and okay, that's it. So as I was saying, the main challenges that we had here was the parameters. We cannot exactly map the parameters because it's a different model. Okay, so and sometimes you have some issues with the paper. Sometimes it was not very clear, and you go to the neuron code, lots of files, and we're like, oh my god, what's happening here? And there were some missing parameter files, and but we did what we could. So Lucas is going to talk about the results properly to you.
Well, so after many trials of different parameters, we could get a very similar dynamics from Sheriff. This is R1. Well, we can see that the PV interneurons are oscillating in a gamma rhythm, and the OLM and pyramidal are in theta rhythm. That is when the OLM are inhibited by the medial septum, the pyramidal starts to fire, and this firing, in addition with the return of the medial septum input, activated the OLM and it gets inhibited again. So this is with the normal scaling. And some difference we get from the original paper is that their PSD was much cleaner in gamma frequency. They have just one peak at 38 Hertz, and we have some peaks here, some around 55. So our high frequencies are less structured than from original paper. When we did the scaling, we can see clearly that, well, as expected, when the NMDR receptors get more sensible to pyramidal firings, so they inhibit more and the network becomes at the limit. The OLM's neurons fires for everything and the pyramidal stops firing. This is also a very similar result from the paper. How this is a transition between a too much excitation regime to a inhibited regime. So we should expect that the optimal values for information flow and coupling should be in an intermediate value. So when we try to replicate the plot from Sheriff, well, in our modulation index, we get a very similar behavior. That is this kind of inverted U shape. So the logic is that when you are in the psychotic regime, you get low coupling between the waves, and if you minister some drug, you can get higher values, but not too much. And but our difference is in the NTE that, well, this is the mean of 30 simulations, and it has a very, very high variability. So we think that maybe it's because of the destructuration of the high frequencies in our model, but we can conclude that we have some level of drug use that could restore modulation index without losing the information flow.
So our conclusions are schizophrenia dynamics might be driven primarily by circuit level connectivity, and this is just a hypothesis. We need to test it better. And synaptic time scale rather than just detail. This is dendritic, but it's neuron morphology. Reproducing the network dynamics NS is possible, but we need to recalibrate the parameters, of course, because a HT neuron is different neural model than the sheriff model. So the parameters should be 2, but we can do it well, like our replication was good. And we also could replicate the findings of modulation index, but the NETA no, and we need to understand exactly why. And yeah, that was the conclusions. And thank you.
I just wanted to give a special thanks for Pedro and Cecilia and Hans because they really helped us to do this project. So thank you, guys.
Questions? So you're basically saying that dendritic morphology wouldn't be as important to this phenomenon that you observe this inverted U shape than the network topology or the timing of the spikes. I mean, what would be the role of dendritic topology then for dendritic processing in this?
Yeah, I don't know. Maybe the NTA that went wrong for some.
Sorry. Well, for me, a big difference between our model and the original one was that they have just one peak in gamma frequency, so it's more organized. And in ours, we have like oscillations, and maybe this regularity is somehow helped by the structure of the pyramidal cells. I don't know.
Have you tried characterizing single cells? Is this so your peak at high frequencies? Is it due to? Could it be due to a single cell property dynamics of a single cell? It's a network mechanism, I think.
We don't know. This is measured for the whole network, and yes, based on the LFP. Maybe if we calculate LFP for smaller sets of neurons or single neuron, we could find something, but we didn't have time to do it.
So yeah, it's a form of computation to see if each single set or single point neurons actually have similar dynamics to the complicated ones.
Thank you.
Hi,
I cannot believe 1 month is over. So in our, I am Oni, Yago and Ariane. In our project, we proposed a new method to do statistical inference in neuronal complex data that preserves complexity. And we validated this method on the critical spiking neural network artificial data. So, human brains and animal brains have hundreds and thousands of neurons, and they have immensely complicated structures out of higher order interactions. And what, as neuroscientists, we record is this. That is, can we make the slides a bit, yeah. Maybe we can go to the presentation mode. Thank you. So what we get as neuroscientists is this, where we have only a limited number of neurons or electrodes, and then we have these undersampled recordings both in space and time. And what we want to do as a theoretician is how to model the unknown. How, starting from this hugely undersampled data, we can obtain a distribution that represents the actual neuronal complex activity. So in this case, it will be like we have these co-activity patterns and different statistics on that, and we want to gain the true distribution. So the easiest way to do it, to follow a principle that says, don't assume anything. So the law of epistemic neutrality. And this is called maximum entropy principle, max-int. What we do is, in this phenomenal paper, the important work from Bialec's group, they did that they have this spiking neuronal data, and they time binned it. They counted the number of spikes in each bin and found out the firing rate activity at the population level, and found out the empirical distribution. And they showed that if they shuffled these neurons, how individual neurons as an independent neuron behave is very different from the population level. So this work shows that population code exists. And the simplest way we can do this population level modeling and try to estimate the distribution following maximum entropy principle is to assume a Ising model, where this population level activity, that is ST, a variable over time t, that changes from one time to another time, and we assume this single j and h, and we want to fit our data on this to find out these are the patterns. So we have this distribution, and we want to fit it and guess the parameters of a very simple model. This is what maximum entropy principle in essence do. But the problem is that we lose complexity. We have only this very low level structure in the model, and when we are fitting, we cannot possibly capture the very complex interaction patterns that are present in the true data that we could not sample fully. So is it good? Is it bad? Let's take this example. So we have this complexity entropy plane, and we have theoretical upper bound and lower bound. And take this picture from Amaril. It has important semantic structures, so it's quite complex, but it doesn't have many components, so the entropy would be low. But then take this picture, this rectangular blue block from Klein, and of course we can see that entropy is very minimum and also complexity. Then take this picture from Pollock, and of course there can be thousands of combinations of these colorful dots, so entropy is very maximum. But by this combination, can we get many different pictures from a semantic point of view? Probably not. But then this beautiful picture from Mars by found by this Peruvian artist Yahu R. Kani, and this picture has immensely detailed semantic structure, complexity. But what maximum entropy principle is doing is basically trying to go to a maximum entropic curve without any constraint. And in essence, what it is doing is to reduce this picture to this. So we are losing essentially the higher order structures present in our neuronal biological data, which could be phenomenologically very, very important. So our proposal was that can we do maximum entropy without changing the complexity? So if our data is here, can we push our data to this theoretical upper bound of entropy without reducing the complexity? And in essence, we keep the data at a complexity level that will preserve the interesting phenomenon. So step one that we decided to do was this biological neuronal network that generates artificial data. And step two that we wanted to do is to compare the vanilla maximum entropy principle to our proposed complexity preserving maximum entropy principle. And we will see what we get.
So we needed a model for that. And no, I'll stay here. We wanted some network that had some biological realism and, of course, criticality. So we first thought of Brunel, but we found pretty fast that it does not have criticality, so we could not use that. But we found a network that was analogous to Brunel and actually extended his network. It is an excitatory inhibitory network of mean field percolation type, which means that we have this excitatory neurons that excites themselves and inhibitory neurons with a strength of J, and inhibitory neurons that inhibits themselves and excitatory neurons with a strength of G times J, but we can also say like WI. But the cool thing about it is that it is an intrinsic stochastic leak, integrate, and fire model, which means that we start diverging from the Brunel model in this thing because first, Brunel in his equations and model, he has to add an external noise to cause sparse spikes. And it is believed that this disrupts the observation of a critical point. And we don't need that because it is an intrinsic stochastic model. So here we have this phi function, and which means that for a time, for a certain time, we have a probability of spiking or not. So here, like below this threshold, it does not spike. Above the threshold, it spikes. But in between, it can spike or cannot spike. So the idea it is that here we have lazier neurons, and this laziness is this slope here that is we get from a gamma variable. So the idea is that their model is not deterministic as Brunel. And this is very important when we consider the ratio between excitatory and inhibitory neurons. In cortical literature, it is 80-20, excitatory, inhibitory. So if this is how it works and how our brains work, there must be something that keeps some sort of balance, right? Because otherwise it would be chaotic. Totally. So the idea is Brunel thinks that there is a balance point. So the inhibitory neurons have 4 times strength than the excitatory neurons. And we have this synaptic balance that is equals 4. But in this model, they consider this laziness of the neurons, and we have this correction term that uses the gamma and other stuff, basically. So here we have, and then we can calculate a critical point that for these parameters is 1.5, but if you change parameters, it's going to change the critical point. And below that, we have supercritical states, where you have more excitatory activity than inhibitory, and the other way around and the other side. So here we have the parameters that we use. We actually only varied network size, but we ended up not using the higher cohort. We recorded neural quantity because we wanted to do subsamples, and we also adjusted the synaptic balance, or this point, to create critical, subcritical, and supercritical states. These are the examples of the networks that we created, and we can see some criticality in them, but it is important to notice that power laws can be presented structurally in a non-critical system, and also limited sample can interfere with critical behaviors, but this is theoretical proven that has criticality, and we wanted to try on it first, basically.
Okay, so since we have a simulated network, we have some data that we can apply to our method. And the important thing about that method is that we can simulate real problems, because we are simulating a full-size network, but we are subsampling it. So we are taking just part of the data, just as we do with brain measures. We cannot measure all the brain. So the EEG needs to be a part of the brain, not all the brain. So these methods allow us to do some predictions about how real data should work. So one of the first things that we can do theoretically is to put one more constraint in maximization of entropy. So this is the model of maximizing entropy in normal research. And we are adding this term as another constraint. So we are putting the model that just like the energy of the system, the complexity should be kept. So we cannot lose energy, and we cannot create energy, but we are proposing that we cannot create complexity and neither lose complexity. We are analyzing the data without losing anything or without creating anything. So the model is more accurate for complex data. So this is a very huge equation to solve, because you have to minimize it, maximize it, and it's very hard, and it's have no analytical solution, because there is a lot of nonlinear terms, but we can approximate it on the regime that we have the log of py, which pi is the probability distribution function, is approximately equals to ln of py plus 1 over n, where 1 over n is the uniform probability. So if you do this approximation, we can look only to the core of the distribution, just the p-values that is very probable to happen. So when you do that approximation, we can look at how the behaviors, the main behavioral shape should look like. So when you do that, we found that formula that have a nonlinear term here that is proportional to how far it is from a uniform distribution. So if this thing is 0, it is just the other, it's the usual maximal entropy model. So we are proposing that it should be a shift between the current model and our model, and we can see that the temperature should be a little bit swifted. So our hypothesis is that that swift goes closer or faster to the critical point. And if it's closer, then we show that with methods we are able to analyze better critical and complex phenomena as a more complex approach. So this is how we can measure critical phenomena in that type of data. Once you fit the parameters, we have an energy function that it is proportional to, it's driven by the easy model. So you calculate the specific hertz of the distribution using a fixed beta parameter. So you fit the beta, you set beta equals to 1 and fit the other parameters. And then we can generate the data like beta equals to 1. And we can see what happens if you cool the data or if you heat up the data. So we are proposing that if you heat up the data, it's like you are giving more importance to small fluctuation, and if you cool the data, you are giving more importance to to more general behavior. Um, yeah, yeah, stable stuff. So this is what about sampled data? Then, okay, so this is how people measure complexity or criticality with that type of model. They are subsampling the model and seeing how the specific head is going to. But one of the interesting things that every time that I show this curve to somebody, they say, oh, it is biological because it's very simple. So it was one idea that motivated us to propose another methods that are not so simplifying the data. So then we calculate the results to a subsample data, and this is our results. In red, we see the maximum entropy curve, just like the previous slide, but it is in log-log scale because we can visualize better, so it may be more flat, but it's not. It's just a way of being. And this blue thing is our curve that it shows that in subcritical system, we have more a cool behavior that it's more flat, and then we, as far as it reach the critical point, in that it become more like a phase transition, and then it completely disappears to the supercritical parameters. So it is the same data, it's the same model. So it is showing that at the beta or at the temperature equals to 1, our model predicts a more close behavior to the critical point. So the critical points in the data should be like this in the peak, and in the maximum entropy model, current model, it should be more like this or this or this. And we are seeing a very more peaked curve, so it's more characteristics of a critical behavior. So with that model, we are leaking how criticality depends on complexity, because once you keep the complexity, the critical behavior is more evident. So this is how we found it till now. We don't interpret a lot it because it finished running 10 hours ago. But thank you all for the attention. Yeah.
So we just wanted to say a special thanks to Cecilia, to Mauricio, and to Fernanda, and to Bialek too, that helped us. Yeah, we're gonna miss you all. Thank you.
Have very short time for just one question. How much of this proposal was done before you came here? Your C backend model, was it done here? You had the idea here, or you applied, or you came with that? Can you? Yeah.
So the idea came from him, a well-developed idea, but then the work was not done. What we did from the
Yeah, it was like I told you, it's like it was a hypothesis based on the oversimplifying figures that we see in analyzing real data. So I had this hypothesis because it's a very common figure in my lab, but I never work with that. So I start to work here, and yeah, I have to ask all the people how to do that in the school.
Did you try to apply it to real data instead?
Yeah,
of
I
just
also
simulate-
apply to real data, and we see the same behavior, but the real data is very constrained data because it's a rat, and they are very, I don't know how to say, they are sleeping or something. They are anesthetized, yeah. So it's a very constrained, and we prefer apply to the model because we can control better the parameters.